until now, we've largely been considering the theoretical side of experimental design. In this session, we're going to talk about some of the more practical considerations that come up when you'll be designing your experiments in actuality. Our point of this session is not to tell you to now throw away all the theoretical design principles we've already learned, but rather to show you that sometimes there's a balance that needs to be struck between what would be ideal from the perspective of randomization and statistical analysis and what's possible in principle. We'll also discuss in this session how sometimes the practical constraints mean that an ideal design can sometimes even introduce more error, even if it's possible to employ in practice. So let's look at some of the potential unintended consequences of an ideal experimental design. So early in this unit, Steve used a football match as a metaphor for an experiment. And I'm from Canada, so I'm a fan of NHL hockey rather than football. But one of the things that struck me when I first moved here 10 years ago was that at a football game, the supporters are divided into home and away sections. And this isn't the case when you go to a hockey game. The fans are all mixed up together. So here we have a hockey game between two of the better known teams in Canada, the Montreal Canadiens in red, which this is a Montreal game, so most of the supporters are in red, and the Toronto Maple Leafs in blue. And if you have really keen eyesight, you'll be able to see some blue supporters throughout the stands. Now in Manchester, we also have a red team, Manchester United, and a blue team, Man City. One thing that we know is that the probability of scoring a goal, whether that's a shooter in hockey or a kicker in football, is that facing a supporter can influence the probability. So if you're facing your own team's supporter, you're more likely to score. And if you're facing an opposing team supporter, you're more likely to miss. So in football, when the supporter sections are separated, we account for this bias by switching which team is at each end after the half time. But what if we instead employ the Canadian model, where we randomize the football supporters in the stands to remove this bias rather than switching ends? So like I said, this is the normal situation in hockey. This poses no problem. Fans of either team tend to get along really well, even though they're bitter rivals, and can be in close proximity without much issue. In football, however, we know that disagreements between supporters are much more common, and putting supporters of two teams closer in proximity could potentially bias our outcome. If the supporters clash, the players could get distracted, or the match could even potentially be abandoned if things get too out of hand. And we would consider this to be a failed experiment because we don't therefore get a measurement of which team is better than the other. So what looks like an ideal design from one perspective, the hockey match, we randomize the supporters, the fans, might be a bad design in the context of a football match. And okay, this is a bit of a tongue-in-cheek example, but it just goes to show that there's no such thing as an ideal design that works perfectly in every situation. Proper experimental design aims to either reduce or control for noise in your data. And that's the variation that's not attributable to one of the treatment effects you're interested in, the residuals term in your ANOVA table, if you like. Now, the point I'm trying to make is that sometimes a design that would be ideal from the theoretical or analytical point of view is actually not so ideal, as it can have the unintended consequence of actually adding more noise into the system rather than controlling for noise we can't control for. Two simple examples of this might be if your experimental design means you need to handle an animal for longer, it could get stressed out and then display some physiological change that's nothing to do with your treatment. Or say if you're randomizing treatments or subjects in some complicated way, you might simply make a mistake. You put the wrong treatment in the wrong place, it gets the wrong label, and then the data from that sample gets misassigned. So therefore you increase noise because you've actually got the wrong label associated with the data. So it's important to never lose sight of the fact that with design, we're trying to eliminate noise. You must consider whether there needs to be a balance between the ideal design and the practical limitations of your system, or even of yourself as the experimenter running the experiment. So now let's look at a more realistic example from my undergraduate days, in which I took a summer field course in fisheries biology. So I grew up as an angler and I'm really fond of fish. And I'm especially fond of these two species. These are freshwater species, commonly called sunfish. On the top, we have the bluegill sunfish, Lepomis macrochirus, and on the bottom, it's the pumpkin seed sunfish, Lepomis gibosus. These two species co-occur in the same bodies of water, but they tend to have different habitat preferences. The bluegill prefers a shallow, weedy, warm habitat, and the pumpkin seed prefers to be in deep water with a stony substrate and therefore cooler water because they're down deeper. They can, however, often be found at the same sites within the same body of water, even though they might prefer to be on one side or the other. Also important to note is that both species are territorial and they will keep to a home range, which they'll usually return to if disturbed by, say, being chased by a predator or caught by an angler and then released. So in this team of undergraduates, we really wanted to ask a simple question about whether a sunfish is more likely to return to a preferred site than to a non-preferred site. So we hopped in a boat in a team of 3 and were set out to sample the sunfish in this lake. And we had a permit to do this from the Ontario Ministry of Natural Resources. So this isn't just something we did on a whim. We were given proper permission to do this from the ministry. So we had our scientific question and we needed now a mark recapture design that was going to answer this question. So the idea here is we sample the fish, we visit weedy and stony sites within the lake. We measure the site characteristics such as water temperature, the depth, and the amount of weed cover. We capture our fish, we mark them using a technique called dorsal spine clipping. So it's a bit like the keratin in your fingernails, it grows back, but it doesn't grow back so quickly that we can't detect the clip over several days. So we mark our fish and now we release all of the fish at the same time. We were thinking here, we don't want to bias our results by releasing fish at different times of day. So we're going to collect all the fish, bring them to a spot in a lake, let them go. So things like daylight doesn't affect their ability to navigate. And then the next day, the plan was to return and see if our marked fish came back to the site where we originally found them, or if they returned to some other site or stayed put. So it seems pretty straightforward, or so we thought. What we failed to account for was that keeping fish in the transport tank for too long allowed the oxygen levels in this tank to drop, which really stressed out and killed a number of our fish. While we did release these fish, we didn't use the data from that day's sampling because of the potential for methods to have biased the data that we got out. Maybe we've selected now for only the strongest fish, whereas the weaker fish might not have come back. So really, we wasted a day's of work and sadly killed off a number of our study organisms. In hindsight, it would have been far better to use a block design to account for any experimental noise introduced by releasing fish at different times, because ultimately we would introduce more noise into the experiment by stressing and killing the fish.